So when it comes to DNA recombination, this is a way that organisms can acquire a new genotype. And so there are four types of DNA recombination, but three of these types are what we call horizontal gene transfer. Horizontal gene transfer is when a DNA or gene is actually being transferred from organism to organism within the same generation. So, for example, you and I, we have what's called vertical gene transfer, where we get all of our DNA from our parents. And so if there was a mutation in their DNA, we would inherit that. If there was a mutation in our DNA, it would be inherited by our progeny. But in this case, it's DNA inheritance within the same generation. So it would be like a mutation in your DNA you could pass to your brother or sister or your classmate sitting next to you. And these three types are conjugation, transduction, and transformation, and I'll talk about the fourth type of DNA recombination afterwards. But the reason why we care about these types of DNA recombination, these three, so much is because of antibiotic resistance. Organisms produce antibiotics that they have created throughout eons of evolution. And we are applying antibiotics for a non-natural purpose, for our medical purpose. We've exploited these organisms, these bacteria and fungi that make antibiotics. And so the bacteria are actually able to swap genes, antibiotic resistance genes, so much faster than the eons and millennia that it took to actually evolve the production of antibiotics. All right, conjugation. You actually know about conjugation. Conjugation is when two bacteria come into direct contact. And are these bacteria gram-positive or gram-negative? Gram-negative only. And what's formed between the two organisms? Called a pilus. And the organism that is the donor that actually forms the pilus has to have what's called an F plasmid. And that's where the genes are housed for making the pilus. Now, there are two types of conjugation. You don't have to know the difference. What you do need to know is that this is the organisms that come in direct contact, gram-negative only, that a pilus must be formed, and an F plasmid must also be present. All right, so conjugation. Here comes a video, and it's going to be loud, just so you know.
Conjugation is a mechanism of gene transfer that requires direct contact between donor and recipient cells. A plasmid is a small piece of DNA, separate from the main chromosome, that carries genetic information for such things as antibiotic resistance. The first step in plasmid transfer is contact between the donor and the recipient. The pilus of the donor cell recognizes and binds to specific receptor sites on the cell wall of the recipient cell. The plasmid then becomes mobilized for transfer when an enzyme cleaves one strand of the plasmid at a specific nucleotide sequence, called the origin of transfer. A single strand of the plasmid, beginning at the origin of transfer, enters the recipient cell. Once inside the recipient cell, a complementary strand to the single DNA strand is synthesized. When donor and recipient cells are mixed together, eventually all of the cells become donors.
So here's another type of conjugation.
Sometimes the F plasmid becomes integrated into the host cell genome. The host cell is now referred to as HFR, which stands for high frequency of recombination. The HFR is able to transfer some of the host genes to the recipient. The sex pilus contacts the recipient F- cell and pulls the cells together. The donor chromosome is transferred as single-stranded DNA starting at the origin of transfer. Gene 1, which is closest to the origin, is transferred first. Segments of the integrated plasmid are at the beginning and the end of the DNA being transferred. It is theoretically possible for the complete genome and the F plasmid to be transferred to the recipient cell. However, this does not happen because the donor and recipient cells will separate prior to the complete transfer of the donor chromosome. The transferred DNA becomes double-stranded. The donor DNA is integrated into the recipient cell's DNA by homologous recombination. The recipient now carries transferred genes, but remains F-, whereas the donor cell remains HFR.
I'm so sorry this is so loud. I wish I knew how to fix that. But the point of these are, we already talked about, they must come in direct contact, gram-negative only. A pilus must be formed, F plasmid must be present, and now we can also learn from this last video especially, we can have anywhere from a fragment all the way up to a whole chromosome being transferred. So of the 3 types of DNA recombination, this one theoretically could transfer the most DNA from one bacterium to another. So that was conjugation. The other type of horizontal gene transfer that we already learned about a little bit was transduction. Transduction is the one where a bacteriophage abducts the DNA and transfers it from one cell to another. There are 2 types of transduction. There's general and there's specialized. What's pictured here is general transduction. So if you remember in the lytic bacteriophage cycle, this is the one where the DNA immediately becomes transcribed and translated and replicated into new bacterial phage, which eventually explode the cell or lyse the cell. Now, the bacterium that has been hijacked during this process is not maintaining its own cell. It's been hijacked, so it's not replicating or transcribing and translating any cell wall components to help those stay nice and firm. It's not transcribing and translating anything to maintain its chromosome. The cell is disintegrating. Everything is all speed ahead towards making more bacteriophage. And so in this frenzy of bacteriophage replication, as the chromosome falls apart, it is very possible that you've got an empty nucleocapsid and you're just shoving DNA in there, and instead of shoving in bacteriophage DNA, you shove in bacterial DNA. And this is called general transduction because there is no rhyme or reason as to what piece of DNA gets shoved in there. But in this case, it's going to be a very small piece of DNA, especially compared to conjugation, right? Because a bacteriophage is so very small, right? Viruses are ultra microscopic, that this is going to be the least amount of DNA that gets transferred from one to the other. And the same is going to be true for specialized transduction. So specialized transduction is, if general was for the lytic phage, specialized is for the lysogenic phage. This is when the DNA gets incorporated into the host chromosome. And when the DNA gets excised and new bacteriophage get replicated, that excision sometimes isn't quite perfect. Sometimes it leaves behind part of its own viral DNA and takes part of the bacterial DNA instead. Now, in this case, every single copy then of that nucleic acid gets inserted into a new bacteriophage. Those bacteriophage are still able to infect a new cell because they still have all their pins and spikes, and so did the ones on the general. But the DNA sequence for the viral for hijacking the cell actually is incomplete, and so that bacterium just gets new phage DNA. Okay, here comes another loud video.
Specialized transduction involves the transfer of only a few specific genes from one bacterial cell to another by means of a phage. The lambda phage, which infects E. coli, is a well-studied example of a specialized transducing phage. When lambda phage infects E. coli, the phage DNA enters the cell and then integrates into a specific site on the host chromosome. When an E. coli culture carrying the lambda phage is induced, phage particles are produced. On rare occasions, a piece of bacterial DNA, for example the GAL gene, near the specific site of insertion remains attached to the phage DNA, and a piece of phage DNA is left behind. The phage that develop are defective because they do not carry the entire phage genome, but can still infect other cells. The defective phage can attach to another bacterial cell and the DNA can be injected. Both phage and bacterial DNA now become integrated into the new host chromosome. Only bacterial genes located near the site of integration of the phage DNA can be transduced, hence the term specialized transduction.
Okay, so do cells need to come into direct contact for transduction? No. It happens via a phage. So what is required? A bacteriophage. Could it be gram-positive or gram-negative? Absolutely, as long as it's the right host, right? It has the right host receptors. Let's see, what is the maximum amount of DNA or how much DNA gets transferred? This is going to be the smallest amount of the three types. All right, so that was transduction. Transformation is the other one. All of these T words. Transformation is when microbes are actually able to take up naked DNA from the environment. So what happens is this naked DNA binds to the surface of the microbe. It gets pulled in and it gets incorporated into the chromosome. And when it gets incorporated into the chromosome, the bacterium actually makes the complementary strand for it. And the person who determined that transformation was actually a real thing, that bacteria could actually take up naked DNA from the environment, was Griffith. Griffith did these experiments with cells that were that cause pneumococcal pneumonia. So Streptococcus pneumoniae, it's a gram-positive diplococcus, and it produces a capsule, and the capsule is part of that pneumonia response. And so living organisms that have a capsule, living Streptococcus pneumoniae, when injected into the mouse is going to develop pneumonia and die. And true to Koch's fourth postulate, you need to be able to re-isolate the infecting agent from the test subject. And so when Griffith isolated this particular Streptococcus pneumoniae, he could find the same organism. He could even tell that it was encapsulated on the petri dish because if you remember, capsules hold moisture, and so the colonies tend to be very mucoid. Now, part 2 of his experiment that proved that transformation was real, that bacteria could take up naked DNA from the environment, was using a mutant organism. Oh dear. Was using a mutant organism that was a non-encapsulated Streptococcus pneumoniae. So it doesn't have the gene for the capsule, but it is alive. And of course then when you infect the mouse with a living organism that is non-encapsulated, the phagocytes of the mouse can easily engulf and destroy that bacterium. And if you can re-isolate, you're going to get very few colonies because most of them will have been destroyed, and the ones that you do get are going to be non-encapsulated because the original infecting agent wasn't an encapsulated organism. It didn't have the genes. It was a mutant. Step 3 of his experiment said, okay, I'm going to take those original cells from part 1 and I'm going to kill them with heat. So he has dead cells. They have the capsule, so they should be able to produce pneumonia, but they're not alive, so they can't replicate. The capsule's falling apart, the phagocytes can easily break this all down, and the mouse remains healthy, he doesn't die, and of course when you re-isolate from that mouse, they were dead in the first place. There's no spontaneous generation. Nothing's going to grow. The fourth part of the experiment, after we've laid out our three tenets, is what really shows that the organisms are able to take naked DNA up from the environment and express them as their own, as horizontal gene transfer. What he did was he took the organisms from parts 2 and parts 3. So he had the encapsulated organisms that were dead, could not form or could not kill the mouse even though they had the capsule. And he had living organisms that were mutants, could not kill the mouse because they could not form a capsule. When he mixed them together, what's happening in that tube before he injects the mouse is the living organisms are able to take up the naked DNA from the dead ones. The dead ones, they're dead. They're decomposing. Their cell wall is falling apart. Their chromosome is crumbling. Their ribosomes are denaturing. Their proteins are denaturing. And so those bits and fragments are all open free in the test tube. And the living organisms could take the DNA that coded for a capsule from that test tube, reel it into the cell, incorporate it into the chromosome, and now they can start expressing that gene. Now they have the new gene, which makes them capable of forming a capsule, and the mouse dies of pneumonia. And when Griffith isolates the organisms, from the mouse, he now finds living organisms that are expressing a capsule gene. So they have the capsule gene, they are expressing it, they have a capsule. And so this proves that organisms can take up naked DNA from the environment. Now in the laboratory, we can do this same thing. This happens in the environment all the time. We can actually use this in the laboratory to our advantage. If we want to take an organism and give it new genes, what we do is we give it a plasmid. We take a plasmid and we put the genes in that plasmid that contain whatever we want. And we also probably put an antibiotic resistance gene in there so that we can force it to keep that plasmid. So we grow it on antibiotic plates. And the way that we do that is we use a cell that's called a competent cell. So the way we're going to get the organism to take up that naked DNA in the laboratory rather than in the native environment is by either heat shocking it or electroshocking it. That's going to cause the cell membrane to kind of expand and contract violently and cause the cell to bring it in. Now, bacteria bring in the DNA from the environment on their own. But if we want to force this into a bacterium, we use a competent cell. Competent cells are called competent because we've bathed them in calcium chloride, which will interrupt the phosphate interactions of the phosphate heads of the phospholipid bilayer of the cell membrane. They're leaky, they're delicate. We have to store them at minus 80 degrees centigrade. They don't last very long because they're very unhealthy. You have to be very gentle with them. Only treat them on ice. Don't pipette very violently. You have to be very careful with our competent cells. But we can get up to a plasmid in these guys. And we can do it on either gram-positive or gram-negative cells. And cells don't have to come into direct contact. In fact, they don't even have to be living for this to happen. So those were the three types of horizontal gene transfer. The fourth type of DNA recombination that students often forget is what's called transposons. That's because it's DNA recombination within the same genome. It's when one bacterium actually has DNA that jump from parts of the chromosome into and out of a plasmid. If you think about it, remember plasmids and chromosomes are supercoiled. They're all twisted up on one another. So parts of that circle that if you had held it out like a hula hoop or the letter O, they would never touch. Because they're all twisted up on one another, they do actually touch. And so this is a way to create new DNA combinations within the same genome, within the same bacterium.
Transposons are segments of DNA that are capable of shifting from one location to another. A transposon enters the cell by being carried on a plasmid. A transposon can then move from the plasmid into the host cell genome. A transposon can move from one site on the host genome to another site on the host genome. When some transposons move, they replicate, leaving a copy in the original position. A transposon can also move from the host genome to a plasmid.
Okay, so those are the 4 types of DNA recombination. You have 3 types of horizontal gene transfer, and you have transposons, which is recombination within the same chromosome. And that is it for bacterial genetics and the end of these videos.